Paper is called a two-sample test for statistical comparisons of shape populations. Statistical shape analysis typically provides distance comparisons between individual shapes. The goal of this research is to provide statistical analysis of shape populations. So by shape we mean the shapes of planar closed curves that are invariant to certain transformations. Modeling shape populations will help us address questions like how different are two sets of shapes? Are two sets of shapes from the same population? How are the shapes in a set related to chemical or physical properties? How will changes in process or environmental parameters affect shape populations? Or how can changes in shape populations be used to predict changes in other associated variables? Modeling shape populations instead of individual shapes is required for many industrial, biological, or environmental settings. So if our question is how different are two sets of shapes, and our solution is to estimate the densities underlying these shapes and compare these. So our comparison framework has 5 components. First, we calculate individual shape distances using elastic shape analysis. We perform dimension reduction using spherical multidimensional scaling. We perform non-parametric density estimation using a Fisher or von Mises Fisher kernel. We calculate the Fisher-Rao distance between these estimated densities, and we perform hypothesis testing using a bootstrap approach. So I'm not sure how well these images showed up over there, but these are images of fluorescent microscopes of protein clusters taken with fluorescent microscopes, and then we extract the curves of these clusters. These cellular biologists are interested in studying their behavior as they research Huntington's disease. This is ongoing research we have with the FSU College of Medicine. So here are the curves we extracted. You should be able to at least see those. And we calculate the individual distances between them. Shape space is infinite dimensional, which makes it difficult to define probability densities. So we map to Sn, in this case S2. The points on the hypersphere are naturally modeled using inner products, making the von Mises Fisher kernel an attractive option for our density estimation. And we calculate the Fisher-Rao distance between these densities. One can use the Fisher-Rao distance for comparisons or clustering, and a hypothesis test can be formed using the Fisher-Rao distance as a test statistic. The asymptotic distribution is difficult to find, so we use bootstrapping to perform the test. So here we have some case studies of actual data that we've worked with engineers in studying. Biological cells, this was actually a NIST study, and we use the same data. We compare our method to 2 other approaches, statistical energy method, which uses the distances to do their hypothesis testing, and then another state-of-the-art method, maximum mean discrepancy, which actually uses the points on S2 to do their comparisons. So on the left-hand side of the screen, we show our different experiments. So the first 3 rows in each experiment represent three samples from the same population, and the last row represents a sample from a different population. And so in the top case study, you can clearly see that the bottom row is much different than the other 3 samples. And in the bottom experiment, the bottom row is visually very hard to distinguish from the other samples. And our method, using the Fisher-Rao distance, correctly clusters and makes the correct p-value assessment. However, we do see that in the last case of the coffee beans, the MMD approach has a misclassification and a near misclassification on that 4th set. Then we do a sensitivity analysis. The question is, how sensitive are each of these methods at detecting changes in a sample? So we start out in the first case by sort of stimulating contamination, where we take a pure sample and from one population and start contaminating it with shapes from another population, and say, okay, how long will it take us to detect that contamination? So in the next case, we take a sample and we start simulating from that sample where we have the same mean, but we simulate an increased variance. And in conclusion, our Fisher-Rao distance is more sensitive than SEM at detecting changes in a sample, and it is less influenced by noise than the MMD.